Okay, so I tend to move very fast, so you can interrupt, and you should, because we can spend as much time on any slide as you want. We don't have to finish. It's okay. People often ask me what a lab's about, because my lab's very diverse. People ask, well, what's it about? And I'll tell you what I want to do. This is both a precise and a vague goal simultaneously. I wish to place any microbe I want in any environment I want, doing exactly what I say it should do. I want to persist there. So I want to be able to put a microbe in your gut, and I want it to reduce inflammation, and I want it to stay there. I don't want it to do anything else to you other than that. I want to be able to place a rhizobium at the root of a plant, and I want to be able to mobilize phosphorus to that plant without having to affect the rest of the ecology. I don't want to get into our waterways. So I want to have a way of engineering fitness and activity in a controlled manner. And that's a long and heavy lift. So I'm going to talk to you about what the challenges are in doing that. When I first started in synthetic biology, we had this vision. And I remember very clearly these conversations I had with Drew Endy when he was at the Microscience Institute. We would sit together at lunch and we'd talk about what we were going to do. And we thought we'd try to make everything very predictable. So I was a physical chemist by training. I thought, I think everything can be physics. We're going to make everything with, promoters very well defined, and the UTRs are very well defined. We put them all together and be like Lego blocks. And I think, in truth, there's still a way of doing that. But the problem is, to do that as an engineer, you have to know what your material is. And the fact is, the material science of cells sucks. Excuse me. And our understanding of the environment that they live in is terrible. And the question is, how do we understand that context and what the engineering principles of that context are? And so I've been trying to figure out ways in which I could get to there very quickly, and I think it's important. And I'm going to first start by motivating. We are engineers by trade. That's what we try to do. But you're bioengineers here. I'm a bioengineer now, even though I was a physical chemist. And the reason why is because we like to be use-inspired science. That's what we like to do. And I like to think about who thinks of what is useful. And my favorite groups to do this are two groups, all part of the same organization called the World Economic Forum. And these guys are great because they're extremely highly paid people who meet in Davos in the winter, and they sit in hot tubs and think, well, what's going to kill the world? And they build graphs like this. And I like two-dimensional graphs, again, physicist. So this is the likelihood of this event being bad, happening. This is the impact of that event. And so up here, I think there's a chronic fiscal crisis. Now, back in 2012, that was out here. So we've gotten worse. But to give you this, it's a highly likely scenario, and it's high impact. Now, there's a bunch of these other little diamonds you see on here, which are things that I think of as biology in the loop. We have water supply crises, which I think are partly, everything's partly policy and partly law. You have to give, that's something we have to deal with. But a lot of it is engineering. Some of it's civil engineering, and some of it I think is biological engineering. Climate change, food crises, biodiversity loss, energy prices, and infectious disease are good examples of things that are biology in the loop. Now, the other group that actually meets in Abu Dhabi, and they also sit in hot tubs for some reason, and they take this list and they think how they're going to solve it. In 2012, they thought two things were going to be the big solution. One, oddly enough, was synthetic biology, which I don't agree with, but they thought engineering biology was going to be a big thing. The other thing they thought was information science, and that was the idea that you had to get the information that you needed, infer what you needed to do, and then work to those predictions. Now, the idea here is that once you had, the reason why they thought biology was going to be important is because you could imagine for things like this discovery of novel antibiotic pathways would be important, and that for supply crises, dealing with plants that could grow in brackish water, or new ways of desalinizing could be important. You could have novel fuel production pathways for energy, and even though we've had this disaster with EDI, whereas oil prices dropped and now cellulose ethanol or fuels don't seem so important. I disagree with that in the sense that if things were priced properly, we would in fact be competitive with that idea. So this is partly a microbial piece. It's also a regulatory piece. Just a word of advice: our lives are going to be ruled by our ability to also deal with politics, and we should learn how to do that. Absolutely. Being part of the national lab, we learn this very quickly. It's part of our job. When you're in Washington, you go see your congressman. No kidding. Food crises and things like that are going to be very important. And I think that food engineering is going to be a very big piece of what we do next. But this is becoming, I think, really profound. We're at an inflection point in our integrated Earth system, and I'll give you some sense of these things. We are in what's called the Anthropocene, which means that where the humans are having as much of an effect on the environment as everything else in the world. We have Earth system scale. Land use is changing. It's doing dietary changes. There's transitional environmental degradation, and it really doesn't matter how you cut it. We're sort of at this point in history where our population growth and the way that we eat is changing the way we work in the world. Water, land, health. But as an example of something that's absolutely critical, even though I'm talking to a bunch of people who are mostly medical, I want to bring this up, is that in 2050, we'll be around 9 billion people. That's about where we'll be. When I was a kid, we were 3 billion people. So we've squared. Okay? To support that population, we need to increase our output of food by 60%. 60%. But if we have the predicted temperature rise on Earth, with the exception of a couple of crops, we're going to have a 40% decrease in agricultural output. That's not a fuel issue. It's not a water issue. That's just climate change. And it's a population change. We have to deal with that. There has been this decline in infectious disease based on our antibiotics and other regulatory aspects. There's an increase in resistant disease that's not shown on this graph. We have an incredible increase in chronic disease that's occurring. And largely this has to do with, one, we're living longer, two, we're living in higher density, and three, our diets are awful. Get to the antimicrobial. This is released last year by the World Health Organization. This is to show you that we're getting this huge increase in AMR. And very importantly, there's been a discovery void in new antibiotics that's occurred since before 1990. And you may ask why that should be, given the genomic revolution that we've had and so on. And the answer is it's very hard to engineer these cassettes. It's very hard to identify the pathways that make these antibiotics. We haven't invested in it. And so we need some way of discovering those and implementing those and creating those. Give you some sense, this is something that should have been conquered, well, hundreds of years ago. Gonorrhea. This is all the things we see the blue crosshatch is where we had to increase the amount of third-generation cephalosporin to deal with the infection. So just to point out, gee, everywhere but Africa, oddly enough. However, there's some hope. First of all, our ability to look at the microbial dark matter, which contains a lot of the tools you would need for antibiotic generation, are scaling technologies, meaning that they're decreasing in price and increasing in the amount of product you get out for that price. And that has led to things like this, where you can sequence large amounts of soil or water or you, and from that, discover many, many, many new gene categories. We'll talk about that more in a moment. And you can discover new antibiotics in new ways. This is a very clever little device, by the way. This is a little chip implanted in soil that allows you to grow these otherwise unculturable microbes that make natural products that kill other microbes. Grow them in situ, but determine what they were. And so we're able to, in this case, they discovered a whole new class of antibiotic that exists without antibiotic resistance. So we're at a point where we can begin to scale these things. The other things that we're beginning to discover that I think are very interesting. This is an example where nearly all the geochemical cycles on Earth, biochemical cycles on Earth, there's carbon, phosphorus, nitrogen, and sulfur, are processed through microbes, through a set of proteins that are expressed on their surface that are these multi-component proteins that do electron transfer. Give you some sense of how odd this is. Electron transfer is like a boring discipline in a lot of ways. It's been around since 1900 as a means of discussion. Yet two new mechanisms of electron transfer were discovered in the last decade. How these complexes parse electrons and move them about. And they're absolutely critical for mediating these cycles. So my view is that a lot of our engineering technologies are going to be going in this direction. We do have some political issues here. This is an issue where this is the projections of the annual flux of carbon dioxide into the atmosphere. So obviously we have data up to 2015 around here. And then this is all the different models that different people run, what's going to happen in the next 100. So first things out, the models suck. right? However, we're dumping large amounts of money into these things. What do we have to measure to get this variance down? Because it's going to be a measurement issue. Their models or stuff, and also their processes are pretty poor. I'm not saying about the processes now. Now it turns out that one of the emerging paradigms is that actually these, again, the microbes and plants are doing a lot of the variation here. That there's a lot of abiotic processes here, but there's a lot of microbes and plants that are doing this. What we're discovering is that there's a huge amount of this soil organic matter which is pulling carbon dioxide in and out, and these organisms do this. I bring this up because I'm talking about where our leverage points are for biological engineering in various ways. Let me give you another one. This is an interesting one. This is the World Phosphate Rock Reserve. Phosphate you don't usually think of as a limited resource, but it is. What you're seeing here is all the places we get phosphate. This big red block, which is where we get almost all our phosphate, is Morocco. One source. Here's where Morocco had a political problem, and the cost of phosphate went up. Phosphate is the backbone, by the way, of nearly all fertilizers in agriculture, along with nitrogen fertilizer, right? It's perfectly available in soil if you could get it out of the mineral form. There are microbes that do that, but we don't use them, and so we have to put phosphate on the soil. What if we could use those microbes more effectively? What if we could engineer them more effectively to do that? We could get rid of this thing. Other things you could put into your mind, getting back to health again, we know now that gut bacteria mediate obesity. You can actually take microbiotic mice who are starting with no microbes, feed them the same diet, and they'll get to some weight. You can feed them one microbe, and one mouse will become fat, and the other mouse will become thin. So the microbes can mediate whether or not you get obesity. Similarly, there are gut bacteria associated with autism, gut bacteria associated with cardiovascular disease. And so as we can get control of these populations, we can get control of a large amount of things going on. This is true of ecology as well. And so, for example, we know that there are organisms that change the way that that termites and other things can process cellulose into molecules we can use for other purposes. And people are beginning to pull the molecules out to use them for those purposes, but can we use these communities themselves to do this better? And to give you some sense, there are all sorts of interesting other economic things going on here. For example, there are beetles that live inside coffee beans that destroy a large fraction of the crops. And these beetles can live in coffee beans because their microbiome can process the caffeine into a non-toxic form. And we know what the gene is for that. And can we engineer phage to go in and kill that gene or kill those microbes, and then these bugs won't eat crops? You can't spray them because they're inside the bean. And so on. So my point here is we're at a point in history where we have these very big problems. We have health, of course. We have environmental stewardship, carbon management, and mineral management. And our population is growing. And there's this famous equation, which you shouldn't take seriously, but people actually use this seriously. You shouldn't take it seriously. Where your impact is sort of your population times your affluence times your technology. And so you can either throw money at a problem, right? Either people at a problem, either technology at a problem. Our job is for technology at a problem, right? So I'm going to argue that what we want to do is we want to do prediction, design, and control from microbiome to biome. Even when you're doing a fermentation, something as simple as a fermentation, what happens in your 1 liter flask is very different than what happens in your 100,000 liter reactor. And that's an environmental change. We have to understand those pieces very, very well. Now microbes are kind of interesting, and people who aren't microbiologists don't know these numbers, so I'm going to throw them at you for those of you who are mammals and think you rule the earth. The first thing to realize is there's 10 to the 30th bacteria on Earth. Okay, 10 to the 30th. That's a hard number to conceive of, but it's more than the number of stars in the Milky Way. It's 10 to the 6, 10 to the 20, 20 to 30 in the observable universe. In the gram of soil, there's 10 to the 6 bacteria and about 10 to the 5th species. So they live very close together, and they split up energetic space in extremely fine ways. To give you some sense, there's 10 to 6 people in San Francisco, and each person emits about 10 to 6 bacteria per hour. And so by far, the most intimate and information-rich mode of our communication is bacteria and not language. It's kind of gross, but there you are. The second thing is, so the bacteria themselves, so you have some sense of their size. They have about 4 megabase genomes. Of course, they vary up to 12 and down to a few hundred kilobases, but that's what the thing is. That's in the third genes. And I can store, I happen to have 8,000 genomes on my flash drive in my pocket with lots of room left over, right? So but they've solved the problem. They're a sense, compute, and do system contained in a micron that have solved the problem of living anywhere that life can live. Bottom of the ocean, on the outside of a space station. 10 to the third protein, 10 to the 7th interactions per cell, about 6,000 different classes of protein in microbes. And so we have a great rich variety of ways of solving the problem of transforming the world into life. So what can we do with them? Well, the large part of what my field does has to do with cell factories. And you have some of the best people in the world here. You have Human, you have Yang Su, Chris Rao, others who are working on ways of having cells turn materials into more valuable materials. And just so you know, when things become interesting is when you're producing materials that cells don't normally produce. And you have to completely redo the chemistries of them and redo their physiologies to do that. But one of the goals we're talking about in the field is any reachable molecule from any feedstock. And of course, there's sense, stimulate, and improve complex environments to control their fitness and activity anywhere. That's what I was talking about at the very beginning. So I'll talk about the first steps a little bit. So here's the easy case. This is something we're doing with EVI. So one of our, and I actually, Yang Su's gotten famous for this recently as well. We're doing it in yeasts that survive in real conditions. So these are yeasts that survive. So Brazil's made an economy out of ethanol. And they have these industrial yeast. But in fact, these are discovered yeast. They were discovered in the yeast when they grew in the reactor. And they only subsist in the reactor for a small amount of time, really, before other guys take over. And so we've begun to really be interested in how you determine the publishing dynamics of the reactors over time. So we use metagenomics to do this, and I won't go into that very important. But the important is that this whole process requires that the cells survive in high acid and high heat. They outcompete wild yeast. They can use a variable sugar supply because it's a very extremely, volatile amount of sugars in each sample. And they survive inhibitors in the feedstocks, which are oxidants and other things. And we've worked with Yang Su, and we've worked with other yeasts to figure out what the genetic determinants of these things are to engineer them to make more ethanol. And so we've actually been able to do that. I'll show you some of that later. Here's another example of a futuristic design and things that we're working towards so you understand what our goals are. We thought back in the early 2000s, wouldn't it be great if we could make a really smart drug out of bacteria? And the idea is that when you cut open a tumor, you look inside of it, at the middle, there are bacteria who live there. In fact, this is pretty generic. I can put almost any bacteria into a mouse, and if that bacteria circulates for long enough, it'll end up inside a tumor and live inside of there. And for a number of interesting reasons. And we thought, wouldn't it be great because these guys could sense being there. They could then actuate a system for going into the cancer cell. They could then sense if they were in the right cell and deliver and synthesize and deliver the kill at that location. So it was sense, compute, and do. Wouldn't that be the way to go? And Chris Anderson and Chris Voigt, who were in my lab at the time when we came up with this, and then Chris went to UCSF very soon after we continued to collaborate, the three of us were thinking about how to do this, and we came up with all these different ways of putting these things together. And we thought, well, we imagined, probably stupidly, you'd inject these bacteria into the bloodstream. And we thought, in order to survive in the bloodstream, they would have to have this capsule on them. So we engineered, we took a base E. coli, non-pathogenic, couldn't survive, couldn't hurt you. We engineered a capsule on it. And we said, could it survive in complement-mediated killing and macrophage ingestion and all this stuff? And the answer is it could. Doesn't survive actually in the bloodstream, unfortunately. But it could survive all the laboratory tests. We knew that they got very high density in the middle of the tumor. They got about 10 to the third higher density in the tumor than anywhere else in the body. We thought, what if they could sense their density? And when they sense their density, we could use this quorum sensing system, and we could have them as they increase density, increase invasion of the nearby cell. And we did that by tuning this up so that only at the right density would they actually invade. And we built this invasion module. This is kind of cool. So Stanley Falco at Stanford discovered this molecule called invasin. And those of you who are into this stuff will recognize the molecule. It's this little tube-shaped protein. You can put on a styrofoam bead, and this styrofoam bead will invade tumor cells. And the way that it works is that it binds to beta-1 integrin, triggers vascularization, and it gets inside. So that's great. And this is just showing that for these different tumor cell lines, we can invade. For any normal cell line, we could not. But once we were inside, so we have this bacteria entering the vacuoles inside there. That's not very useful. It's inside this acidic compartment. It's going to be eaten by the cell. So what we did is we figured out how to sense that we're inside there. And once we were inside there, we could break open our cell, break open the vacuole, deliver our kill. That was the idea. And we did that. But we only want to do it under certain conditions. So we built AND gates and logic gates that told us only do it when certain conditions are met and so on. However, these two finally gave up. And they gave up because, I think they're wrong about giving up. They gave up because it's hard. It is hard. There's so much low-hanging fruit right now. Why do the hard stuff? But this is a large system. This is, by the time we get to this point, we had about 250 kilobases of material stuck into E. coli, right? It's a high load on the system. Every single thing we did in the lab, we tuned by hand in vitro. None of that stuff works in vivo. In vivo is a different world. You can't do any selections in vivo. You can't, because it's unethical. So you have to get better design here. So you have to figure out what's going wrong. What's the context issues? Here's something that's actually kind of interesting. This has actually become very interesting. We started back in the 2000s. We thought, could we build a parasitic virus for HIV? And what this virus would do is it would actually, I'd put it into your T cells, and it would hang out there. And then when HIV infected the T cells, it would detect this. It would downregulate HIV, but it would steal HIV's capsid and mobilize itself. And it was cool because when you introduce a prophylaxis into a community, like a condom, to prevent sexually transmitted diseases, the sociology of this is people have as much sex as it takes to overcome the condom, more or less, because they feel safer. And so we thought, wouldn't it be great if we had this therapeutic virus? And they give you this virus, it's a one-shot affair, you don't have any compliance issues or things like this. It sits in your T cells, you inject you get HIV, and what it does is it mobilizes our therapy. So then if you have sex with somebody, which you shouldn't, well, without a condom, you have sex with somebody, you transmit the therapy virus, not the HIV. Sounds perfect. Leonard Weinberger, who is actually at the Gladstone right now building this. He's built models that do the whole epidemiology of it. And this is very similar to what people at the church lab were thinking about in terms of gene drive. And we try to introduce genes into the environment that take over and displace a pest species and prevent them from reproducing. So we're at a point in history. My lab was all computational at this point, and we actually built this damn thing in the lab. It took us 2 years. We're at a point in history where we can do this. So as engineers, what's our ethics? I would argue that what makes engineers is not getting the effect we want, it's getting the trust to implement that effect. I can get a paper airplane to fly, but I won't get you to fly on it, right? The point is, you have to do both. Engineers engineer for effect and reliability. And if we're going to be doing synthetic biology where I want to do it, which is inside of you or in the soil or in the water, you have to trust me. Or we have to try it out and it doesn't fail enough times that you trust me.
That involves the whole regulatory process, the whole how do you?
It does.
How do you see going through that?
I think what's important is, first of all, you're going to get some successes. A good example is Carl June at Pennsylvania, who did the adoptive immunotherapy, the gene therapies, and got slapped down for it hard in 1992. This guy was you guys don't know Carl June, you should know Carl June. Spectacular guy. Wonderful set of stories, too. Heartbreaking. He just does that. He just does stuff. And then when the need is acute enough, people let him do it, right? And so, no kidding. So in a lot of cases, you have to just do it. But I think that as I said in the very beginning of this talk, one of the things I've learned as part of the national lab, because we do spend a lot of time with government, is you can be extremely effective. An individual can be extremely effective when they go and talk to OSTP. You can just call them up, Office on Technology Policy. Call them up. You can go and talk to this guy when you go to Washington. You can talk to your congressman staffers. This is extremely important. As universities, speaking as a department chair, we can go and we can present these things, and that ends up in the national discourse, and I think we should. But we have to deal with that thing. Now, the point of context is this. I'm going to show you that we're going to try to map context in an efficient way. And I'm going to say there's a bunch of different types of context here. We talked about them in these two papers, so I'm not going to belabor the point, but just to say the following. There's basically these 4 types of context that I'm going to talk about today, maybe only 3. One is environmental, which is I want to know how differences in the environment impact how my cell functions. But I don't want to know it just from a physiological point of view. I want to know it from a genetic point of view. I want to know how every gene plays a role in allowing my cell to grow and perform some function in a given environment. I want to do this over a large number of environments such that I can make a map that says this gene is important generically because of these effects and these environmental parameters. So we need to build a model for gene-host interactions, which are gene-environment interactions. There's also host-gene interactions. As I put a heterologous pathway into a cell, I want to know how every host gene affects it. Why is this important? Well, it's important because even in cloning strains of E. coli, if I put the same pathway in cloning strains of E. coli, it's entirely different. But you are different from everyone else. And so your genetic differences, when I put something into you, are going to impact how my thing works. And I need to understand that host-circuit interaction. And finally, there are other things here in molecular interaction. When you put two things together, or two molecules interact, there's context interaction. We'll talk about that as well. Let's talk about environment first. This is a map of Bear Valley in Tennessee. It happens to be where we've made nuclear weapons. And so there is uranium, which has a gradient across this valley roughly this way. There's nitrate, and there's technetium, and there's phosphate, and there's different pHs, and it's a bloody mess. And it's not good. But a lot of waste got contaminated. Everything you see, all this blue is, of course, waterway. It's a watershed, and water is moving out towards Oak Ridge Town down that way. And all these little holes are holes that we've drilled into that site. And we look down in the holes, and we ask what's going on down there. And so some people look down and we'll ask, how much uranium is there? And we have around 150 to 200 different parameters that we measure about each hole over time, not well over time. And we've been doing microbial assessments. And so what we do is we took a set of these holes that varied these key biogeochemical parameters in a factorial design, statistical factorial design. So we had these 100 wells, all of which vary these parameters. And we then did metagenomes, which in this case are not whole genome shotgun, but they're taxon, they're amplicons. And we asked how different bacteria classes changed as a function of these variables. And we were able to build interaction maps that sort of told us how every microbe was constrained or not to different environments. And we could figure out there are guys who only like low pH, and guys who like high pH, and guys who like only high pH, but only when there's high sulfate, and so on. So you now have this environmental map. We then bring them back to the laboratory, and we simulate those environments in the laboratory. So we have these miniature simulations. This is going to be a repeated thing in bioengineering, which is having small-scale simulations of natural environments. And we're still struggling with this here. But we're looking at groundwater, so we have groundwater. And we then vary these parameters in groundwater. And we grow them in these things. We grow them in these 96-well plates. And we developed the technology that allows us to, by sequence, measure the population dynamics of every single microbe in a complex consortia over time and over all these things very, very cheaply, relatively cheaply. Which means that I can do hundreds of 96-well plates. So about a few plates per lane of alumina, more or less. And I can measure quantitatively the population abundance. And then I can do things like this. Now this happens to be 6 microbes in a community measured across communities which are changing their pH and their salt. And what you're seeing here is their population abundance at an endpoint. So they would have different dynamics at an endpoint. So what you're seeing, for example, is that Castanella really likes low pH, low salt, whereas Acidovorax likes high pH, high salt in this particular case. And you can see that they kind of they have some correlation. Now, we've been able to do exometabolome, we did temperature and exometabolome, and we can show that in each one of these cases you get different metabolic intermediates, and you've been able to map this in the metabolism of the organisms and figure out how they're dissipating that energy. And so now we have a map of metabolism to abundance over time and temperature and different variables. And then we can then build models and we can figure out how different organisms would interact. For example, in this case, B. cereus really does not like Rhodonobacter, and it kills it dead. And so we're now being able to figure out what are the key environmental and cell-cell interactions that permit these cells to live in a given environment. And how does that hold over many, many environmental parameters? To be able to pull out, by factorial analysis, what the key determinants are that make that so. Now, once we have that, we want to figure out what the genetic reasons for this were. The problem is that unless you're E. coli, even if you are E. coli, when you have E. coli, you know a rough annotation for 60% of the genes. You know a good annotation for less than 50% of the genes. So when you look at annotations, we have, if you look across known microbes, this is based on a detailed analysis of 20 chosen and hundreds of randomly sampled microbes, you have very good annotations for about 20% of the genes. You have OK for an additional 40%. but very poor and close to worthless for the rest of them. If you go to a microbe like I cut it from the soil, I might have half of that good, and everything else very poor, close to worthless. That's where we are. So we have to get better at that. I won't talk about that right now. So we're trying to, we use 3 technologies to link genes to phenotypes quickly. And I'm going to talk about only 2 of them today. I'm going to skip this one because Yang-Su can talk to you about it some other time. And that's how to use high-throughput insertional mutagenesis, barcoded, or CRISPR-I manipulation to query these things. The way both of these things basically work is you have reagents that you can affect the functioning of some region of the genome. So with a transposon, it inserts a particular region. And what we do is we have these transposons barcoded. They can only insert once. Where we infect the population, and we infect at such density that we get a population that has strains with the transposon. There's at least one strain of the transposon at a given location, such that across the population, we would transposon every 20 base pairs. So by sequencing, I can now say, this transposon is this insertion in the genome. And now I have 15,000, 20,000 strains, each with a different transposon in a different location, different barcode. And then I can use sequencing to ask how much of that barcode is in a population. So if I have a population of these strains, if one wins, its barcode gets very, very high. Or if one loses, its barcode gets very, very low. Okay, so it's barcoded sequencing. And we've been able to do this over this year, 20 bacteria over hundreds of conditions. Okay? So here's 1 year's work. We did 100,000 transposon insertion mutants. We've done strain fitness changes over time, 4 to 6 generations, typically contain 20 mutations per gene. And we've done phenotypes for 18,000 genes. This is more than has been done in the past century. Okay? And these are true genetic variations. We've done 17 bacteria, 11 genera, 5 divisions, 2,000 different assays with all these different stressors and carbon sources and things like this. And we were able to group genes by operons and things like this, and we were able to find these very clean situations. You have 19 carbon sources, triplicates each. You know if a gene is important. In this case, if you don't have the gene, you die. In this case, if you don't have the gene, you live better. So the first thing you realize is not having the gene is not necessarily a good thing. Okay? You get to this point where it's extremely consistent. So we can take, this is a pure biological replicate. These are actually 2 different weeks, and we can get this very nice correlation between the fitness for day 1 and day 2. And we have consistency in 17 bacteria. This is these are in defined and stresses. And you can say linear correlation of replicate experiments, and you can see we're all the way up around 0.9 or so. So it's doing pretty good here. Now, what this allows us to do, because of the way that we design the experimental conditions and these transposons, is we've been able to get we've been able to get informative phenotypes for example, 1,500 of 1,900 genes in Shigella, as an example, or in Xenomonas. So we can get nearly the entire genome out of this. And these are informative phenotypes, are ones where I have some idea of what the biochemistry of that particular protein is. That's a huge sea change from where we are right now. What's really interesting is you can figure out how much a given gene has confers a phenotype under all conditions. So for example, this is a particular gene where if I knock it out, in 80 conditions it has a phenotype. Whereas these guys over here, this histogram, these guys over here only had a phenotype in 1 condition. So most genes are very specific, which is one of the reasons we don't discover them. Some genes, essential genes, are everywhere. So we have some measure of pleiotropy now. We know what that distribution looks like. And it turns out that this is the probability of different categories of genes having a phenotype. That every category of gene has a high probability of having a phenotype. Even guys who are orphan genes, guys who have a domain of unknown function, or actually are not even called as genes, some of them. This then shows the distribution of some of these things in different microbes. And so we just did a bunch of these things. We found that we could really begin to annotate a huge amount of these different microbes. And again, what we find out is that we can take even things that have had no annotation in literature previously and assign them a particular function. Not necessarily an EC number with 4 digits, but all of them. And even guys who are paralogous have function. That is, different paralogs of the same gene have different function. Now, why am I telling you this? I'm telling you this because this gives us an opportunity to do large-scale annotation across the tree of life in microbiology. We've done this in fungi, archaea, and microbes. What it also gives us is a chance that I can do this across many, many environments. So now I have a way of looking at environments and genes in a clever way. Let's not go through all of this stuff. This is just a bunch of quality control. But this is important. If we now take that just the 17 organisms we did this last year, and we ask if you take random sample of other bacterial proteins from the databases, which don't have annotations, and say, can I now give them an annotation based on our experiments just this past year? We have, for about 30%, so the more similar they are to a homolog, if they're very, very similar, we can give them a 40% chance of giving them some data. We can actually give them a biochemical phenotype in about 20% of the cases. If they're even very divergent, we do have some chance of giving them some annotation. I'm going to give you some idea of what this allows us to do. This is a case where we had a strain of microbe that could survive in ionic liquids. Ionic liquids are a way that you can deconstruct cellulose biomass directly into sugar and lignin, where lignin drops out, so you don't get sugar. And it's very complete. But the ionic liquid is poisonous to the fermenting bacteria. So we thought, wouldn't it be great if we could find why it was this bacteria could survive in ionic liquids? So what you're seeing here is fitness in ionic liquid, fitness in a defined medium, and fitness with ionic liquid. So these guys up here are genes, if you knock them out, they don't grow anymore. And down here are genes, if you knock them out, they grow better. So we found genes for both. And what we did, I don't have the graph here, so we engineered this gene, this gene, and this gene, and that gene into a bacteria, into a yeast, actually, in this case. And the yeast could then survive in ionic liquids thereafter. Here's the kicker. We have no idea what those genes are. They were one hypothetical, two duffs. No idea. Same thing happened with Xymomonas. We have this. This is again, it's an example of a bug that makes ethanol. Same exact thing. We had many, many conditions, many, many genes. And the first thing that came out is when you cluster all this fitness data, you can find out, as you do with other types of assays, that the genes cluster by their chemical behaviors. So you can assign chemistry to the clusters into the genes. And then we could take those genes and we can begin to engineer them into, re-engineer them in Xymomonas to produce more ethanol. So in this particular case, this is wild type, this is an engineered strain. And the fundamental idea here is that it's sort of sad. Again, the thing that worked was two hypothetical genes. All the ones that we thought were going to work didn't work at all. So the first thing it teaches us is that the genes that are not annotated yet are extremely important. It's unfortunate. And two, we need better ways of getting to those genes. But three, you can in fact engineer based on this knowledge of how genes confer fitness in different environments. Turns out you can start to get to general principles this way. We put in expression cassettes into these deletion libraries. We're able to find out, for example, that by looking across how genes get differentially expressed under these library conditions. So this is, for example, two different fluorescent markers which under different knockout conditions express these two fluors at different levels. And we could assign, this is a gene, this is a knockout for a particular metabolic enzyme. And by looking across the classes of things, we found out that one, there were sets of genes that controlled cell size, sets of genes that controlled signal transduction, sets of genes that controlled purine metabolism. And what they change, if you change purine metabolism, you change how variable your cell population is, it turns out. If you change glycolysis and ATP, you change your mean production rate. If you change your genes involved in energy and AA metabolism, you change your cell size. And we can begin to get some sense of how each one of these engineering tools that we're going to use to optimize gene expression would affect the physiology and outcome of our population. Now, there's other things we were able to disprove along the way. There's been a series of papers in the last year that have put out that environmental microbes are auxotrophs. That is, they require materials made by other cells to grow. And we've been able to disprove that entirely. The reason why people think that is because they couldn't identify the genes that did the amino acid metabolism. It turns out that central metabolism is far more variable than people thought it was. That there are many non-canonical routes to making amino acids, for example. And so we've been able to find all these protective auxotrophs, grow them in defined media, and show what gene it was that allowed them to grow in that situation. And we've done that by using that sort of technology. We've been able to fill lots of holes in biosynthetic pathways. The figures here aren't important, just to say that we can do it. We fill in catabolic pathways. We've identified bogus annotations. And give you some sense, for given microbes, say E. coli, we actually came up with 193 new annotations. And we demoted 50 in E. coli. Crazy. We've identified a novel DNA repair nuclease that occurs in the periplasm. There's something interesting in the periplasm during DNA transfer. And the other thing we discovered was that phenotypes are not conserved. So if you take two Shigella species and you ask how their genes, if two genes are co-fit or have the same phenotype under the same conditions, you find out that even though they're strict orthologs, they do not contain the same functionality in a fair number of cases. Not all cases, but a fair number of cases. So what we're beginning to discover is that genes are repurposed very rapidly in the environment. And we're figuring out how they're doing that. Let's just go to this plot. This is a better plot. Yeah.
So how much is known about this methylation issue here?
For the DNA, you mean? Right. So of course that's true. So actually the interesting antibiotic story turns out that of course methylation, especially outside the gamma, is incredibly important for cell cycle regulation, but it's always been important for restriction of DNA transfer between cells. That's where restriction enzymes come from. And DAM methylases and things like that are things that methylate DNA so they can't be cleaved by the restriction enzymes. They're also signals. They prevent transcription factors from binding. However, JGI has done a global survey of methylation in microbes, and it's a weak signal for most of the physiologies we're talking about. It's very interesting. It's very important. But Lucy Shapiro, working with Steve Benkovic from Penn State, has discovered an entirely new class of antibiotic that prevent the DAM methylation from working, and is extremely effective against firmicutes and things like that. So very interesting. Okay, phenotypes not being assured. So this is an example where this is fitness in ANA1, ANA3, and density if it's fit in MR1. So here here, for example, if the fitness is very, very bad in ANA3, then it's also very, very bad in MR1. But as you move up the spectrum, become less strong phenotypes, these are like nearly essential. You can see that basically there's no correlation. They're all around 0. Okay, I believe at this point enough. I'm going to stop there. There's another way of doing this. Now that's all about single genes. What I just told you, though, is that when you take a gene into a new context, into a new host, its phenotype changes. And so that means that phenotype is conditional on the rest of the genes in the cell. And actually, its biochemistry is as well. That I won't go deeply into that. And so Stanley Chi, when he was in my laboratory, when he went up to UCSF, and now Harney Rishi have been working on this topic where we use these oligoarrays to build guide sequences for Cas9. Does anyone here know what Cas9 is, how it works? You all should. It's basically an RNA-directed endonuclease. So it's the RNA guide sequence will be bound to Cas9. Cas9 will bind to the complementary piece of DNA and cleave it. In this case, we prevented it from cleaving it. And instead, the Cas9 just sits down on the DNA and prevents polymerase from processing or other proteins from binding. And so we built an array that tiles over the E. coli genome, so we can have all these guide sequences for the E. coli genome, and we built a library of them. And that allows us then to use these guide sequences, these Cas9, which I can use the transposons, and I can downregulate every gene in the organism and see how its phenotypes are. So this shows that I have here's 3 genes, here's 3 guide sequences, and in this particular condition, all guide sequences behave the same and they all go down and so on. And in the end, we can do experiments like I just showed you, where you have aerobic growth and anaerobic growth, and find genes that are only important and essential for anaerobic growth, as an example. And it's nice because this is inducible, so wherever I build the library, it doesn't matter. Now, because guide sequences are small, I can make arrays of them, or I can put many of them on the same plasmid. In this case, I put 2 of them on the same plasmid, and I barcode them. So now I have this plasmid which has guide sequence 1, barcode 1, guide sequence 2, barcode 2. I can put that into the thing. Now, what this allows me to do is do epistasis. I can ask, how much does this phenotype change from the single mutants when they're both knocked out? And for those of you who don't know what epistasis is, if you can define it this way: if the phenotype is bigger than the sum of the phenotype of A and B, then it's positively epistatic. And if it's less than, it's negatively epistatic. So it turns out that, of course, there's many variants of these things. We built in such a way that we could pull out subgroups. So, for example, all small RNAs are an 18,000 variant library, and all cell division proteins are about 108,000. Transcription factor and binding site variants are 3.5 million variants. So we have a lot of different, these are big libraries. Turns out we can take that entire population, take 100,000 variants, for example, with 50-fold coverage and 5 million reads, and run this whole thing on a MySeq. So it turns out that I can do the big libraries on a High-seq. So I can read them all up by barcode sequencing. And so what we did here, this is actually fairly hot off the presses, where now we have, this is guide sequence 1, guide sequence 2, all mapped to where they map in the genome. And then if it's yellow, there's no phenotype. If it's red, it's a very strong phenotype, and if it's blue, it's a very strong positive phenotype, very strong negative phenotype. So you're seeing here now is I can say, for example, this is an example to show that it works, that there's a huge, that if you take these two different proteins, DNA C and FITZ, both involved in sort of chromosomal replication and cell division, they have fairly strong defects, and if you add them together, it's exactly the same as a double, right? So no epistatic interaction, individual functionality. However, if you take FITZ-L and this conserved protein with an NADP binding domain, which has no other annotation than that, these two, this one has a strong effect, this one has no phenotype, put them together, it's almost as strong as the pair above. Strongly positive. And we also have examples of strongly negative as well. So we're beginning to get this epistatic map. There's also beneficial ones. This is the positive ones where here's two guys who almost have no effect alone, but together have a huge effect. Again, note that one of them is a Y gene, meaning it has no annotation. So the first thing we're beginning to figure out now is we now have a map that has all gene pairs. This gives us now networks of genes, all of whom interact together. So what we've been able to show you here is I now have a large-scale, I've used large-scale synthesis, rapid assembly and technology, and barcode sequencing to allow efficient genome-scale querying of genome function. I can grab annotation of these genes, and it guides multiplex engineering for complex phenotypes. So I can do these fermentation phenotypes for these genes. Now let me go a little bit deeper for a moment. There's some future bacterial gene annotation here. I think we can really begin to use this technology to blow through annotation. And I'll pitch the fact that we can do a lot of this work now in this other project I run called the DOE Systems Biology Knowledge Base. For those of you who are interested in taking genomes to metabolic models, this is a fantastic platform for doing that. It allows you to collaborate with others. It allows you to share your data with your laboratory. So I use it. But we're getting to the point where all the technology I'm telling you about, all the tools for analyzing that data are being brought into the Systems Biology Knowledge Base, and all the reagents are being sent to AdGene. So you guys can all do it here as well. It's all being open sourced. Okay. Let's talk about this now. I'm going to talk about this next level here. Let's talk down to the other level. So one piece of context is how environment, gene, and host interact. I just showed you that. Now I'm going to talk about compositional context, where things have to interact together. The idea here early on was that we'd have DNA. We wanted to get central dogma in place where we could control DNA to RNA to protein, and we understand this stuff. And the idea was that we'd build these cassettes, and we'd have these different parts, the promoter part, the UTR part, and the CDS part, and we'd do this. And you'd build libraries of each one of these topics. And then by knowing how each one of these things interacted, you could predict what would go on. And so what we ended up coming up with is this idea that we wanted to have these libraries of principal boundaries. And if we could do this, we could do things like this. You could take a promoter and a regulator and another regulator, and it would change the way the polymerase or a ribosome moved down the nucleic acid. And because it occurred in order, the function that you'd produce was a multiplication of the individual regulatory functions, right? So you could build any function this way. That was sort of the idea. Oops, something went wrong. I think it's okay. I think, given the time, this is probably okay. Yeah. Let's see how it goes. So at the time we did this, we recognized another thing, which was that even when we started doing our assays, all our assays were relative. And even if we could get them to work sort of predictably relatively, we couldn't get them to work absolutely. Because it would be absolute levels. So we started thinking, what's the problem with this? We started thinking that maybe translation was the problem. So we built these libraries that allowed us to put a 96 nucleotide encoded peptide upstream of SFGFP that would be cleaved off when this was translated. And we'd vary this to vary how translation worked. And so the idea would be that we were going to test how every nucleotide in here played a role in changing the quantitative behavior of this cell. So that's what it encodes is SFGFP and its cleavage product. We're going to read out which of these 96, which of these strains we have by barcoding, and we're going to be able to control ribosome procession and binding and so on. So, yeah, okay, so we can skip the whole part of this stuff, that's fine. So the idea was that we took these hypotheses. There were 8 biophysical sequence properties that people had thought were important for translation, as an example. And so it turns out, 1,158 combinations of these 8 sequence properties. We did these in 3 variants each, so triplicate, in 56 different regions of sequence space. Okay, so now we have this huge library. It's literally 244,000 sequences, 1,000 sequences in 56 different regions of sequence space, each testing 8 different biophysical theories. We then cloned this library into E. coli, did DNA synthesis by adjuvant array, cloned it, and put it into E. coli. And we measured 5 things: protein growth, protein production, cell growth, mRNA, mRNA decay, and polysome load. For every one of those 244,000 mutants, all in a one-pot assay. Okay. So the first thing we found out was that we made sure that there's the difference between sequences in a series was very low, between series was very high, both at the nucleotide level and at the amino acid level, so that they really are very distinct regions. And here's what we found out. First thing we found out was that when you did analysis of variance, because we had this factorial design, we found out that, so first let's do some coding here. We varied, for example, the structure around the UTR and CDS. We varied structure sort of into the gene and further into the gene. We varied the AT content. We varied the hydropathy of the amino acids being encoded. We varied the codon usage, other things. First thing we found out was that we barely explained 24% of the variation in the expression with these things. So that means most things were not explained. There's a great deal of error, but a lot of stuff was not explained. Most of the explanation was due to structure around the UTR, rather than these other things people care about. And most of the error was due to the different regions of sequence space we were in. So there's some common mode failure depending on what region of the world we're living in. If you take one of these guys and you look for a given series, one of these 56 regions of sequence space, that's what the bar looks like. So what you're seeing here is, this is the explainable effect size for protein production. This is for all of them. And in each series has a different series of factors. These are all the factors we tried. And you can see that red dominates that structure. But it's not always the most influential. And it's not always the majority. So it's very different depending on where you are in the sequence spread. And almost every other paper in the area used one sequence and varied it very small. First thing you realize is that biophysical theory was not looking everywhere. So this violates that. So now we can go ahead and tamper with that structure. What we're going to do is we're going to build a system. This is built on based on other technology in the laboratory where we can put a leader sequence upstream of this ribosome binding site here. I think I do it. This leader sequence here, and we have another ribosome binding site over there. The ribosomes bind here, and they move through this leader sequence, translating it, unwinding the RNA, because they have helicase activity. We can stop the ribosome from doing that by putting a stop codon there that is suppressed by using an unnatural amino acid. So I can turn on and off structure. So when I turn off structure using this technology, you can see all the red goes to the bottom. So not that we get rid of it, but that now other things become important. And it turns out that the structure 5' of that region becomes very important at that point. One thing to take away from this is protein production, a histogram for each one of these series. And you see they're very, very different from each other here. Whereas back here, these are crazy different from each other. We have guys who are dead and guys who are, extremely high in protein production. So they're very different. So in the end, what we were able to show, since we're at the end here, is that there's a huge trade-off going on in the system. And that while it's mostly structural, there's a complex nonlinear interplay of these parameters and much of we don't know. And one of the things that comes up is that translation, that the RNA itself is mediating cell energy, even this one piece of RNA. So what we did is we measured growth rates by just measuring the DNA of each of these constructs and compared it to the RNA of each of these constructs. And so we get plots like this, where we have protein production, versus relative growth rate. And you can see the following. One is that if you follow the median here, it'd just be this. It's largely flat up to a critical value, and then you begin to die. Okay? This is because you're beginning to use a lot of resource, making a lot of amino acid, using a lot of amino acid. But you see this curtain down here? That has a lot of RNA in it. So you have RNAs that can kill the cell without producing a single protein. Now, because of the time, let me just skip to. I think we can do this. Sorry. I should be able to do that here. I can't do it here. I'm going to get out of this for a second. Get this thing here. There. Okay. So this is just that same plot. You have it's sort of hard to see because of the lights, but you have protein, and you have fitness, and it goes this way. I've colored it by the structure of the 3' CDS, so you can see, for example, that the structure, which is in the middle of this 96-well region, it's yellow on the top, so these guys have higher fitness. Yeah, we're good. Yeah, that's fine. Yeah, good. So you can see that there's these visuals. So you have this yellow is higher fitness, for example. But this whole thing is actually a wing in three-dimensional space. So what's interesting about this is that despite the complexity of interactions, what we're determining is that while none of the biophysical theories fit this curve, there is some physical theory to be had. It's sitting on a wing. Now, there are guys down here which I don't understand. Don't get me wrong. But most of the density is sitting on this wing. And it turns out that every single physical parameter localizes on this wing. Let me show you a split through this. So here, for example, this is that wing I was just showing you. This is a density plot now as opposed to showing you every single point. And this is just saying that, look, if you have protein and RNA, the guys who grow best are here. If you have protein and growth, the guys with the most RNA are down there, not up there. If you have most RNA and you have the most growth, well, and the guys with the most protein are over there. So you have to figure out what you have to engineer to get things to work. So this is something which we're mapping all those things. Now, what we were able to do is to map the polysomes to this thing, and this is the important take-home message. What you're showing here is how many, what the sequestration of ribosomes are on RNA in the cell by this measurement. What you're seeing is that this is deciles, I guess, deciles, or maybe 20 iles, duodeciles of sequestration. And so this guy is the most sequestered. And you can see he's killing. These are transcripts that sequester ribosomes and don't release them to make protein. So they're killing the cell. These guys are guys who just make a lot of protein and sequester a lot of ribosomes. And they die. Here's guys up here who release ribosomes very quickly, and so they don't die as much. So we're beginning to figure out what all the costs are there. So this is the cost of production on this. We can sort of deconvolve this. This is cost of production. And this is cost of using the factory. So this is actually cost of materials, amino acids and things. This is cost of ribosomes and polymerase. We can quantify that in various ways. So the take-home message of this is that the two things I just showed you we're able to use hypothesis-driven functional genomics for consistent quantification of effect sizes, and explode this out into lots of new information about cells very, very quickly. That was this last stuff for one student's work, Guillaume Cambrai. We're able to, in this particular case, figure out that the structure is the most important thing. That structure has particular characteristics that either sequester or don't sequester ribosomes. And we're going to figure out what that is. Codon usage modulates elongation, but only when all structures are taken care of. And that there's a lot of nonlinear associations here that require us to do some modeling. And in the end, as a whole, what we're getting to is the ability to do these very large throughput screens to come up with engineering principles that allow us to design transcripts and to design cells to interact in these complex environments in better ways. So that'll stop and just thank the team. I think mostly I want you to know Guillaume Cambrai did that last part, and Adam Deutschbauer and Jeff Skirker and Morgan Price did the first. So thanks.
talk. Question? Yes,
Okay.
So this transposon-based method and this CRISPR, I think both methods have pros and cons.
Yep.
But the biggest problem is whenever we introduce this mutation, you said you found two hypothetical proteins, but the gene causes interesting phenotype. And the problem is suppressor mutations.
Yep.
Right, so are you sure, did you sequence other genomes? Are you sure that there's no kind of unknown mutation happening?
Yeah, we do. First of all, the way these are built is we control for the number of divisions they have to go through. So they should be very identical. We always reintroduce.
I see.
So we can always do the direct knockout assay. In some cases we do the direct knockout assay. In other cases we can do the complementation assay. And in both cases it works. So in these particular cases that I showed you, we actually use them for engineering. And so the gene itself worked as expected. When we resequence the genome, which we've done many times, we don't see any other mutations. There are cases, however. I should mention. You're right, obviously. So if we take our best performer out and we let it grow for even a small number of generations, it turns out something else is toxic, and so you get mutations in the genome. And I'm not sure why. I'm not sure how to deal with that. In this case, we only let them grow for 4 to 6 generations, and so we're looking at the rate of rise, and so a late mutation wouldn't show up. So it has to be in the existing population to begin with. Yeah.
And also, the transposon is kind of blind for the essential genes.
It is.
Right, so that's why I put the
CRISPR's not. We see all the other genes.
of CRISPR.
Actually, you want to hear something really annoying? So that's true in E. coli. It turns out that in Xenomonas or in cyanobacteria, of course it's not. The transposons can insert two essential genes because there's multiple genome copies.
Ah.
And it turns out that's extremely useful. But CRISPR does this itself anyway.
But still, this transposon and CRISPR require these genetic tools.
They do.
Like at least one transformation method like that.
Yes.
But we have tons of microorganisms do not have those tools.
Yes.
So the idea is just to random kind of, we create a library by random mutation or evolution and then the sequence.
You can, but the density is too low. And so to do what we're doing, so transposons, we have a library of 10 transposons now with different transformation elements that we've used, and we are having an 80% success rate at environmental isolates for getting the transposons in. So it's not so bad. Not great, but not so bad. We have a new method though, and it's a small molecule. That is a minor groove binder, and we're able to program that in. We're hoping maybe that will be the way to go next. But I agree that genetics holds us back. For the fungus, it's taken us 2 years. We only got our first libraries last week. We now have full genome scale libraries of all the agenous yeasts. Using agrobacteria mediated transposon mutagenesis, but it's a pain. Yeah.
Any other questions? Yes.
So I was interested when you were doing the gene pairs and one of the genes had an annotation, but one of the other ones may not have.
Yep.
And you were looking at sort of their cooperative effect. Could you use that result using one annotated gene and or I guess several annotated genes and one non-annotated gene to infer the annotation?
Yeah. So that's actually the major use of the tool is we use those epistatic effects and co-fitness effects to assign an annotation. And one of the big questions that we have, right now we do it very bluntly. It's sort of naive Bayes, and which so you take the probability of it having that and just sum them up and that's it. Yeah. It turns out that overpredicts, and so we need a better computational methodology for doing that probabilistic annotation. And we have people looking at that. Actually, Sergei Maslov, who is coming to UIUC, is one of the guys who's helping us do that. Yeah.
All right. So because of time limit, so for the last step, so in brief appreciation of Arkin work, so Department Head, Raj would like to
Very formal. I'd just like to thank Adam and very busy man, took his time to come here, really appreciate it. And just a small token
Oh,
of
thank you so
appreciation,
much.
along with the rest of your awards, you can put that on.
Anything, sir.
Thank you.